It's titled The Structure and Function of Large Biological Molecules. And our focus of this chapter is going to be to look at some of the very large molecules that you do find inside a typical cell. Now, most of the molecules that we look at in this chapter, they will be what we call polymers. Polymers are simply large molecules that are constructed by hooking together many smaller subunits, which we call monomers. This is going to be very similar to a chain that is made up of smaller links that are all identical to each other. When that chain is put together, those links are hooked together one after another until we end up with maybe thousands of them hooked together and a chain that is now many feet long. We do have specific reactions that will be used to construct these macromolecules or polymers, and the reaction that's used to actually construct them or put them together is going to be what's called a condensation reaction. Now, this is called a condensation reaction because there will be the loss of a water molecule, or H2O, when this reaction takes place. So another term that's used a lot of times to refer to it is a dehydration reaction. And I like that dehydration reaction term because it is a little more descriptive. Now, if we look in this example, we have this short little polymer. It is made up of 3 subunits. We can see here we've got 1, 2, 3. And on one end of this polymer, or one end of this chain, we're going to have a hydrogen that's hanging off. Now, this is certainly a simplified diagram because what's shown as the purple circles would have larger structures underneath it, so there would be other elements and atoms inside there. But we're going to focus in on this end where we have 1 hydrogen hanging off, and then we're going to have a smaller subunit which we call a monomer, mono because it's only 1, and it has a hydroxyl group, which is the OH group hanging off of it. That hydrogen and the OH are going to be removed, and when they are removed, they combine to form 1 water molecule. In the process of that, the number 3 subunit is going to get hooked to this new one, and the new subunit will become the 4th monomer in this chain. Now, we could repeat this process again if we had a need to. For instance, we could take another smaller subunit that had a hydroxyl group hanging off of it, remove another water molecule, and this could then become subunit number 5 hooked on there. So when we're building polymers, it's not a complicated reaction. It is just a dehydration reaction where we remove a hydrogen and a hydroxyl group. They combine to form water, and now we've got this molecule that is 1 longer than what it used to be before. So if we were, for instance, to hook together something that is 400 monomers long, that would require us to perform 399 dehydration reactions. Notice how we're going to have basically 1 fewer reaction than we actually have monomers. That also means that in that particular case, we would remove 399 waters to actually form that. Now, not only can we put together polymers, but we can also take them apart. One example of that would be a starch molecule. Starch is a huge polymer, and when we consume starch, which is quite a common food item, our bodies perform reactions that take apart the monomers so that we can then get energy from them. So the reaction that's going to disassemble or take apart the polymers is what's called a hydrolysis reaction. The term hydro, you probably are already familiar with the fact that that means water, and lysis, which is a new term to us here, means splitting. So what we're essentially doing is we are splitting these links that we just formed by adding a water molecule back. So if you look at this figure here, we are now going to take that water molecule, we're going to force it into that link, and when we do, the hydrogen attaches to one side, the hydroxyl group attaches to the other side, and we have now broken that link apart. If we were to actually run 3 hydrolysis reactions, we would completely break apart this polymer. So we would add one water molecule in at each one of the links. You want to make sure that you're familiar with the type of reaction that is used to assemble polymers. That would be the dehydration reaction. You want to understand that in that case a water molecule is removed. And then we also want to know the reaction that's used to take these macromolecules apart from each other. So that would be the hydrolysis reaction. And in that case, we're actually forcing a water molecule back into the links to actually break the subunits apart from each other. Now, one of the reasons we're talking specifically about macromolecules is because macromolecules are extremely important molecules to living organisms. Some examples of polymers would be carbohydrates, also known as sugars, proteins, and then also nucleic acids. And nucleic acids, if you haven't heard that particular term before, this is DNA, and another example of it is RNA. So I'm sure most, if not all of you, have heard of DNA. If we look at a typical cell, within a typical cell, 70% roundabout is composed of water. So that is the vast majority. But that leaves us about 30% that is going to be chemicals. And of that 30%, most of it will be macromolecules and most of it will be polymers. So here we've got on the bottom, we can see that we've got polysaccharides. That is a type of sugar. We do have proteins there, RNA and DNA, which are nucleic acids. And then there's also going to be some small molecules in there. So other than the water that makes up the inside of the cell, most of what we have left is going to be macromolecules, and most of them will be polymers. The rest of this chapter, we're going to look at examples of macromolecules in detail. We're going to talk about the structure of those molecules and some of the individual properties that they have. It is important by the time you finish your studying of Chapter 5 that you are able to recognize examples of carbohydrates and proteins and the other molecules that we do go through. So along the way, we'll be talking about the characteristics that you should be looking for to be able to identify these. The first type of polymer or macromolecule that we're going to talk about is what we call a carbohydrate. Carbohydrates, if you just break apart that word, it means hydrated carbon. So carbon with some water. And that is essentially what we're looking at in this case. Because it's carbon with some water, that right away tells us what elements we ought to find inside a carbohydrate. We will have some carbon, we will have some hydrogen, and we will also have some oxygen there. Now another way to refer to carbohydrates is to call them sugars. And that's a very common term that you hear in everyday life. Those sugars can also be called saccharides. Now, depending on the size of the molecule, we can call them monosaccharides, disaccharides, or polysaccharides. Mono basically means one, di means two, and poly means three or more. So that's the only difference between them is really how many individual monomers have we hooked together. So we're going to first talk about the monosaccharides, and if you learn how to identify the monosaccharides, then it should not be very difficult to identify the disaccharides and the polysaccharides. So we already said that monosaccharides are going to have the carbon, the hydrogen, and the oxygen there. Generally speaking, if we're talking about monosaccharides, they are going to have some kind of a molecular formula that is a multiple of CH2O. So one water molecule for every carbon that is contained in the molecule. Monosaccharides are not that large. They typically contain 3 to 7 carbon atoms. So here we have some examples on this slide. Notice that each one of these monosaccharides has a short little stretch of carbon atoms hooked together. So that would be what we would call a carbon backbone to our molecule. Now, in addition to the short little carbon backbone, there are specific functional groups that you do find in carbohydrates. The first functional group that we have is what we call a carbonyl group. Now, the carbonyl group has the carbon that has a double bond to an oxygen. And in this particular picture, all of the carbonyl groups have been shaded in orange. Notice that they can be located at different places within the molecule. So sometimes they are located on one of the ending carbons in the backbone. Sometimes they're located on one of the middle carbons. Depending on their location, we can have different names to our monosaccharides. When the carbonyl group is located at the end of the chain, those are called aldose sugars or aldehyde sugars. When it happens to be located in the middle, then they are called ketoses or ketone sugars. You will see those terms pop up in the lab as we go through our classifying carbohydrate lab. So it really is just a difference in where that carbonyl group is located at within the molecule. The second functional group that we always have in our monosaccharides is the hydroxyl group. And there will be lots of hydroxyl groups inside the monosaccharides. So with the monosaccharides, one carbon will get the carbonyl group, as we just pointed out, and all of the other carbons are going to have hydroxyl groups on them. So all the rest of them will have one hydroxyl group each. So notice that there will be a whole bunch of them. And the remainder of the structure is just to add enough hydrogens on there to give every carbon 4 bonds surrounding it. So most of the time, the monosaccharides, when we draw them in the simplest fashion, we draw them in this linear format. We can draw them in a circular format as well, but we want to make sure that we do understand the linear format first. So if we draw a very simple monosaccharide here, just to review, we're going to have a short carbon backbone. So I'm going to make a 5-carbon sugar. So this would be what we would call a pentose because it has 5 carbons. So notice we've got a short carbon backbone, and we really only range from 3 to 7 carbons. So they're not going to get a whole lot larger than what you see on this slide. One of the carbons in the chain is going to get a carbonyl group. So I'm going to put it on this one right here in the middle as an example. Since it happens to be in the middle, this would be what we would call a ketose sugar. But we're going to get one carbonyl group. And the rest of the carbons are all going to get hydroxyl groups. Now these hydroxyl groups could be pointed in either direction because keep in mind these are single covalent bonds, which means that these bonds would be free to rotate around. So while they may be pointing to the left at one moment, they may be pointing to the right the very next. In addition to the carbonyl group and all of the hydroxyls, we need to just make sure that all of the carbons have 4 bonds total surrounding them. And that means that we need to add enough hydrogens onto our molecule to give every carbon its 4 bonds. Notice that in this case, the carbon that has the carbonyl group right here in the middle already has 4 bonds on it, so we do not need to add an additional hydrogen onto there. That's the basic monosaccharide structure. If we were to go through here and figure out what the molecular formula is for this molecule, we would have C5. If you counted up all of the hydrogens, you would find that we have 10 hydrogens, and then we have 5 oxygens. Notice how we have twice as many hydrogens as we have carbons and oxygens. So in other words, this does reduce down to the CH2O formula that we introduced. So 1 water molecule for every carbon that we have. Another thing to notice about this is that there are going to be a lot of polar covalent bonds inside the carbohydrates. Notice that all of these bonds I'm putting an X on, they would be polar covalent bonds. Since they are polar covalent bonds, that means that we would have that separation of charges that we talked about in Chapter 3, where we would have little areas where we have a positive charge and little areas where we have a negative charge. What this is going to be good for is it's going to give water molecules a lot of little areas that they can interact with. This is going to allow water molecules to basically coat the surface of our carbohydrate, or in other words, to make a hydration shell. So our water molecules would surround so that they would have the opposite charges next to what you see here. So remember that the pointed end of the water molecule would be the oxygen end that would be a little bit negative. So that would be attracted to the ends that are a little bit positive. Now, it is true that every bond in this molecule is not a polar covalent bond. So there are certainly some non-polar covalent bonds in there, but when we look at the whole structure, there is a good scattering of polar covalent bonds over the surface of this molecule, and it will give the water enough to interact with to get a good hydration shell on there. What this means is that our water molecules are going to surround this, and this will be something that is then hydrophilic. Okay, so hydrophilic, so our water molecules will surround the surface of it. In other words, carbohydrates are soluble in water. Now, that was a linear monosaccharide that we looked at. Monosaccharides are not always linear. They do tend to circularize, so they can circle around on themselves to form a ring structure or a cyclical structure. We see glucose in this slide doing this, and over here we've got the example of the ring structure to glucose. When you look at a carbohydrate, a monosaccharide in a ring structure, you will see a small ring. This ring is either going to be a hexose or a pentose. So in this example we see that it is a a hexagon or a pentagon. And you'll also see that while most of the corners of the ring are going to be carbons, you will have one corner that is going to be an oxygen. So we'll have one oxygen embedded in the ring, and then we are also going to have lots of hydroxyl groups on here. So if we point out those hydroxyl groups, there they are. Notice that our carbonyl group has disappeared when we formed the ring structure. So you're not going to be able to see that carbonyl group anymore. So just to review that, we're going to have some kind of a ring structure. The ring can be either a hexagon or a pentagon. So here we see the pentagon. And then we're going to still have lots of hydroxyl groups hanging off of this molecule. Okay, so here we've got the skeletal formula shown more, so similar to the formulas we looked at in chapter 4. Just to review those skeletal formulas, remember that each point that we see is actually a carbon, and each carbon would have enough hydrogens on it to give it 4 bonds total. So this one right here would have 2 hydrogens coming off of it. This one would have 1. This one would have 1. Here we have 1, and here we have another 1 as well. Okay, so that would be one example of a monosaccharide in a ring form, or we could have the pentagon, which is what we see there, and there would also be lots of hydroxyl groups coming off. All right, that's our monosaccharides. The monosaccharides, it's important to be able to recognize the structure of them, both as monosaccharides that are linear and ones that are circular, because those are going to be the building blocks for the disaccharides and the polysaccharides. So we can take these monosaccharides and we can start to hook them together using dehydration reactions. So the same type of reaction we talked about at the very beginning here. When we hook together the monosaccharides, it is going to be the ring or the cyclical forms that are connected together. Now here we see an example of 2 glucose monomers that are going to get hooked together with a dehydration reaction. That dehydration reaction is going to remove a water molecule, and when it does, we get the glycosidic linkage that we see named right here. The glycosidic linkage is the link that we form between these 2 monomers. It is always going to have an oxygen atom embedded in the middle of it. And that then is what we call a disaccharide. Notice that the disaccharide is two monosaccharides hooked together with a glycosidic linkage. Now, we can hook together two of the exact same monosaccharides as you see in A, or we can hook together two different ones. But even if we hook together two different ones, the method will still be the same. It will still be a dehydration reaction, and it will still form a glycosidic linkage in the middle. If we perform even more dehydration reactions, that's where we end up with what we call a polysaccharide. Generally speaking, polysaccharides are going to have a lot of subunits or monomers that make them up. Usually we're not talking about just 5 or 10, we're usually talking about thousands of units. And the polysaccharides vary depending on what monosaccharide is used to compose them, and then also the positions of those glycosidic linkages. But we do have a number of examples of polysaccharides which are very important in nature, and some are used for energy storage, and some of them do serve as building materials for cellular structures. So if we look at a few examples of these, one of the biggest examples that we have is what we call starch. You've probably heard of starch before. Starch is an important component of many of the foods that we eat, so of potatoes, rice, wheat, to name a few. Notice that all of those things that we've listed are plant-based products. That's because starch is a storage polysaccharide that is manufactured by plants. What this means is that when plants have a lot of extra glucose or a lot of extra sugar around, monosaccharides around, they hook it together with dehydration reactions to compose this polysaccharide called starch. This provides a very compact way to store that particular carbohydrate. Now, plants do have two different forms of starch. They have what's called amylose and amylopectin, and the difference between the two is really the amount of branching that you actually have within the molecules. If we look at it in just a little bit of closer detail, here you can see the structure of the polysaccharide. Notice that you have many of the monosaccharides hooked together. We can still see those glycosidic linkages here. Keep in mind that each one of those glycosidic linkages would have been formed by a dehydration reaction. And notice we can also have some branching in our molecules. So that's what determines the difference between the amylose and the amylopectin. So what we're looking at in this particular slide, we see a cross-section of the roots of a plant. Roots are very important storage organs for a plant. So keep in mind that that's essentially what a potato is. So potato cells are full of granules of starch, which is what we're seeing in these cells here as well. Now on the animal side of things, animals do not make starch, but they do make a similar product called glycogen. Glycogen is also made by dehydration reactions, forming glycosidic linkages. It is highly branched, and it, as we said, has a similar structure, similar function. Provides a very compact way to store sugars inside of a cell. On the structural side of things, cellulose is the most abundant organic compound that we know of on the Earth's surface. So obviously this one is going to be very prevalent. This is a major component of plant cell walls. Now the cell wall is a structure that is around every single plant cell. So if you can think about all the plant cells that must be in the universe, there's going to be a lot of cellulose. So they're around all the cells of plants. They are also polymers of glucose subunits, but these are going to make very long linear molecules, which make long fibers that make up that very rigid cell wall. Offers a lot of protection to a plant cell. So that's a run through of the carbohydrates and their structures. So just to review, the monosaccharides can be both linear and they can be circular in form. They're going to have carbon, hydrogen, and oxygen. There are two functional groups that you find inside of carbohydrate. They have one carbonyl group, if we're looking at it in a linear format, And then they're going to have lots of hydroxyl groups as well. Now, if we look at them in the monosaccharide form, they are going to have a formula that is some multiple of CH2O. Once we start hooking together the monosaccharides to form disaccharides or polysaccharides, that CH2O formula is not going to work anymore. And that's because each time we form a dehydration reaction, there is going to be one water molecule removed. So we will no longer have one water per carbon that we have inside this molecule. Another structural polysaccharide is chitin, just to show you that there's some variety there. This is going to be making up the exoskeletons of a lot of arthropods, so insect exoskeletons. And this particular one actually does have some nitrogen contained on it. So here we can see that shaded in blue in the monomer that makes up the chitin, just to show some of the variety. Now this slide right here shows you a good example of the type of thing that you should expect on your exam. On the exam, you will be expected to identify carbohydrates, be able to pick them out if there are a whole bunch of carbohydrates being shown to you. So if we go through here and we first pick out the monosaccharides. Remember, with the monosaccharides, if we look for them first in linear form, first of all, they're going to have a short stretch of carbons, 3 to 7 carbons long. They're also going to have one carbonyl group, and they're going to have lots of hydroxyl groups. The first one that we have listed on here would be I. So I would be a monosaccharide. It has 5 carbons, so 1, 2, 3, 4, and 5. We have one carbonyl group here at the end, and then there are lots of hydroxyl groups on this molecule. If you come up with the molecular formula for this one, it is C5H10O5. So this one is very similar to the one that we drew. So that one would be a monosaccharide. Another monosaccharide on here would be P. P is also a linear monosaccharide. The only difference between P and I is really the length of the molecule. So when we look at P, we actually have 6 carbons there. You still have one carbonyl group, lots of hydroxyl groups, and it would reduce down to some multiple of CH2O. This one would actually be C6H12O6. Now in this example slide, we do not have any examples of circular monosaccharides, but we do have some examples of disaccharides. So if we look for the disaccharides are going to take the ring form of the monosaccharides and hook them together, forming a glycosidic linkage in the middle. Our first example of a disaccharide would be J here. Notice with J, we've got a ring that has one oxygen embedded in it, and we still have lots of hydroxyl groups. We have a second ring that has essentially the same thing. Notice as well that right between the two you have a linkage that has an oxygen in there. That would be the glycosidic linkage. So that would be a disaccharide. Another example of a disaccharide would be R down here. R is simply drawn by a different molecule drawing program. Notice you still have 2 rings that have oxygens embedded in them, lots of hydroxyl groups, and you still have that glycosidic linkage there that has the oxygen embedded in it. If you can identify the monosaccharides and the disaccharides, the polysaccharides should not be any trouble because the polysaccharides are just larger. Usually the polysaccharides are thousands of subunits long. We're not going to show you one that's thousands long. So if we see 3 or more hooked together, we want to call it a polysaccharide. B up here on the top would be a good example of a polysaccharide. That one actually has 4 long, although it does continue on in both directions. And another example would be L down here with a polysaccharide. So all of those, the disaccharides and the polysaccharides, do contain some glycosidic linkages. All of them were constructed using dehydration reactions, and all of them could be taken apart if we were to use a hydrolysis reaction.